Hello everyone, and welcome. Today's lecture, BioC 770 Lecture 6, will delve into the fascinating world of oxidative stress and how our bodies respond to it. As you can see from the visuals, oxidative stress can impact everything from the freshness of an apple to the aging process in humans. I'm Mohammed Shahid Nadeem, and I'm excited to guide you through this topic. Let's start with the basics: free radicals and ROS, or reactive oxygen species. Radicals are molecules with unpaired electrons, desperately seeking to fulfill their octet rule. They can act as oxidants, stealing electrons, or as reducing agents, donating them. Remember, all ROS are radicals, but only oxygen-containing free radicals qualify as ROS. Now, what is oxidative stress? It's essentially an imbalance. When ROS production overwhelms our antioxidant defenses, cellular damage occurs. Think of it like a scale. When ROS outweighs antioxidants, the resulting oxidative damage can harm lipids, proteins, and nucleic acids, ultimately leading to tissue injury. Where do these ROS come from? Well, they're naturally produced as byproducts of normal cellular processes, especially during oxygen metabolism. A major source is the mitochondria, where ATP is produced. During this process, electrons can leak and react with oxygen, forming superoxide radicals. Oxidative enzymes also play a significant role in ROS production. NADPH oxidases, OX, and the mitochondrial electron transport chain, ETC, both convert oxygen to superoxide. Then, superoxide dismutase, SOD, converts superoxide to hydrogen peroxide. Xanthine oxidase, XO, also contributes by producing superoxide and hydrogen peroxide during the conversion of hypoxanthine to uric acid. Our immune system also utilizes ROS. Phagocytes like neutrophils and macrophages produce ROS to kill invading pathogens. This process, called phagocytosis, is a primary defense mechanism. Neutrophils can even release their nuclear contents in neutrophil extracellular traps, nets, to ensnare and kill microorganisms. Let's revisit the mitochondrial electron transport chain, or ETC. As electrons move along this chain, oxygen is reduced to water. However, this process isn't perfect, and ROS, like superoxide anion, hydrogen peroxide, and hydroxyl radicals, can form as byproducts. ROS generation isn't solely due to internal processes. Environmental factors like pollution, radiation, toxins, and pesticides can contribute. Lifestyle choices such as smoking, poor diet, and alcohol consumption also play a role. Furthermore, certain diseases like inflammation and infections can increase ROS production. ROS can have both positive and negative impacts. On the positive side, they help kill microorganisms and regulate cell signaling. However, excessive ROS can lead to lipid peroxidation, protein inactivation, DNA mutations, and ultimately, oxidative stress, aging, and disease. Thankfully, antioxidants like enzymes, vitamins, and hormones can help counteract these effects. Lipid peroxidation is a key consequence of oxidative stress. It occurs when ROS attack lipids in cell membranes, particularly polyunsaturated fatty acids, PUFA. This process damages cell structure and involves a chain reaction with initiation, propagation, and termination phases. Vitamin E plays a crucial role in terminating this chain reaction. Lipid peroxidation produces various products, with lipid hydroperoxides, LOH, being the main one. Other byproducts include malonylaldehyde, MDA, propanol, hexane, and 4-hydroxynonenal, 4-HNE. MDA is a potent mutagen, while 4-HNE is highly toxic. MDA is often used as a biomarker for lipid peroxidation of omega-3 and omega-6 fatty acids. To understand lipid peroxidation, it's helpful to visualize the structure of lipids in plasma membranes. These membranes are composed of phospholipids, glycolipids, cholesterol, and proteins arranged in a bilayer. The hydrophilic heads face outwards, while the hydrophobic tails face inwards, creating a barrier between the extracellular fluid and the cytoplasm. Oxidative stress can directly damage these lipid membranes. Mitochondrial ROS can initiate lipid peroxidation, leading to the production of cytotoxic lipid aldehydes like 4-hydroxynonenal, 4-HNE. This, in turn, upregulates ROS production, creating a vicious cycle that impairs cell function as seen in human spermatozoa. Oxidative stress also affects proteins. The process involves an initial state with a native protein, followed by oxidation due to oxidative stress, resulting in an oxidized protein. This leads to modified proteins, modified peptides, and modified amino acids, which can be analyzed to understand the extent of oxidative damage. Sulfur-containing amino acids like cysteine and methionine are particularly vulnerable to oxidation. The sulfhydryl group of cysteine and the thioether group of methionine are prime targets. Elevated levels of cysteine sulfonic acid are linked to type 2 diabetes, while higher cysteine levels are associated with Parkinson's disease and HIV. Aromatic amino acids such as tyrosine and tryptophan are also susceptible to oxidation. Tyrosine, being redox active, can form tyrosyl radicals that dimerize to form dityrosine, a fluorescent protein crosslink. Tryptophan can be oxidized by hydroxyl radicals to hydroxytryptophan. Elevated dityrosine levels have been linked to several diseases, including type 2 diabetes, rheumatoid arthritis, Alzheimer's disease, Parkinson's disease, aging, multiple sclerosis, and autism spectrum disorders. This highlights the widespread impact of oxidative stress on human health. Oxidative stress doesn't spare DNA either. Both the ribose sugar and nitrogen bases can be modified. ROS-based DNA damage can lead to nitrogen-based degradation, single-strand breaks, and cross-linking to proteins. Hydroxyl radicals are particularly damaging, reacting directly with all DNA components. One common form of DNA damage is the formation of 8-oxoguanine from guanine. This can lead to transversion mutations, cancer, altered transcription, and embryopathies. Understanding these mutations is crucial for comprehending the long-term consequences of oxidative stress. Now let's talk about the heroes, antioxidants. These molecules neutralize ROS and free radicals, preventing damage to cells, proteins, lipids, and DNA. They work by donating electrons to ROS, stabilizing them, and stopping the chain reactions of oxidative damage. Antioxidants can be broadly classified into enzymatic and non-enzymatic categories. Enzymatic antioxidants include primary enzymes like SOD, catalase, and glutathione peroxidase, as well as secondary enzymes. Non-enzymatic antioxidants include minerals, vitamins, carotenoids, organosulfur compounds, antioxidant cofactors, low molecular weight antioxidants, and polyphenols. Superoxide dismutase, or SOD, is a crucial enzymatic antioxidant. It converts harmful superoxide to oxygen and hydrogen peroxide. SOD uses metal cofactors like copper, zinc, manganese, or nickel to alternatively add or remove an electron from superoxide radicals. Catalase is another important enzymatic antioxidant. It uses iron or manganese as cofactors to catalyze the degradation or reduction of hydrogen peroxide to water and molecular oxygen. Catalase essentially completes the detoxification process initiated by SOD. Glutathione peroxidase and glutathione reductase work together in a critical antioxidant system. Glutathione, a tripeptide, is a key antioxidant in the intracellular environment. Glutathione peroxidase converts reduced glutathione, GSH, to oxidized glutathione, GSSG, during detoxification, while glutathione reductase regenerates GSH from GSSG. Glucose 6-phosphate dehydrogenase, G6PD, is a metabolic enzyme involved in the pentose phosphate pathway, which is vital for red blood cell metabolism and protection against oxidative stress. G6PD deficiency, a genetic condition, is more frequent in males due to the G6PD gene being located on the X chromosome. The hexose monophosphate shunt, or pentose phosphate pathway, is a metabolic pathway parallel to glycolysis. Glucose 6-phosphate can enter either glycolysis or the HMP. In the HMP, G6PD converts glucose 6-phosphate to 6-phosphogluconate, reducing NADP plus to NADPH. NADPH produced by G6PD is crucial for redox metabolism in red blood cells. Glutathione reductase uses NADPH to convert oxidized glutathione, GSSG, back to reduced glutathione, GSH, which is then used by glutathione peroxidase to detoxify hydrogen peroxide. Thank you for your attention. I hope this lecture has provided you with a comprehensive understanding of oxidative stress and the human body's response.